Mm. All right, good morning. How are we doing? Okay. Uh, this is going to be a more challenge to do it on Zoom, but we've got this. We can do this. Today we are going to be talking about macromolecules. So let me make sure we have everybody here. Participants. Okay. What I would like everyone to do is please, in the chat section, type in that you are here. That's going to be how I'm going to do attendance today. So in the chat. And Drew, you just got in. Make sure you put in the chat that you are here today. That's our attendance. Let's see. Okay. If you haven't put your name in the chat, please make sure you do it. That I know I just said it, but it is our attendance for today. I'm not going to do an attendance question today. So let's make sure you get in for that. Okay. So with that, I am going to go ahead and get us started. If I find the right PowerPoint. Okay. Now I'm going to try to figure something out. So bear with me for just a second. I want to see if I can have the chat while I am lecturing so I can see you guys talking if you're asking questions. If you do have questions, go ahead and unmute and just ask. Okay, you don't need to wait for anything. Just unmute and ask. I will see you or your picture, whatever you've decided to put up there. Okay. All right. Macromolecules. So we're just going to go ahead and get going on this. Okay. The main macromolecules, there's 4 that we're going to talk about today. The first being carbohydrates. Sorry, I'm going to make sure my phone was on silent. Second is proteins. The third is lipids. And then lastly, nucleic acids. Oh, one thing I do need to say, we have to end class a tiny bit early today, which is okay because we are totally on schedule because we actually have time to talk about macromolecules also on Thursday. Hopefully you all got my email, I think you did, about I need to go get COVID tested just to be on the safe side. That's happening this morning, so wish me luck. So that's why we have to end a tiny bit early so I can go and be tortured by that brush up my nose. So that's going to be awesome. Super awesome. Okay, continuing on. Monomers and polymers. So each macromolecule is made up of subunits called monomers. And these monomers, these identical monomers, join together to form a polymer. So if you remember my little story about poly, poly means many. So many means many monomers, okay? Usually this means 3 or more monomers joined together. Okay, so bearing that in mind, we're going to start talking about our first macromolecule, the carbohydrate. Okay, in carbohydrates, we have a carbon, hydrogen, and oxygen ratio of 1 to 2 to 1. And just to remember, try to write this here, I don't know how to erase, C6H12O6 is the base glucose molecule and that is a carbohydrate. And if you look at the numbers, it is a 1 to 2 to 1 ratio. Okay, and that equation, C6H12O6, is one that I would expect you to know. Okay, that you would need to know that equation, that is glucose. This is going to come up again when we talk about metabolism, so it's something you will need to know throughout the semester. So might as well just learn it now. Okay, monosaccharides. Some examples: glucose, fructose, and galactose. I feel like maybe this is did we start this last time? Well, it's okay if we did. Oh, wait, what do we got here? I think we have some people trying to join. Give me 1 second. Where is it? Okay, we got people here. Don't need that. Sorry, 1 second, guys. Everybody's in. Okay, sorry. Thank you for bearing with me on that. Okay, so glucose, fructose, galactose, these are all simple sugars, essentially. They're carbohydrates. They are a single monomer of a carbohydrate. And you can see that they look very similar. Fructose is a little different because it's a 5-carbon ring instead of a 6-carbon ring like glucose and galactose. You do not need to know the actual image, okay? You don't have to be able to write it out, for example. These are just just so you can see what they look like. If there is something that you have to identify or be able to write out, I will let you know. So glucose is one of those things. Okay, oh, kitty cat. Do we need to have pet time, guys? It's okay, Drew. She's cute, or he. I'm sure at some point you'll hear my dog bark. Oh, he turned his camera off. It's okay, Drew. It's okay. I actually like seeing you guys' faces. Okay, disaccharides. Okay, this is when you have two or more, oh, I'm sorry, two, di means two monosaccharides that have been joined together by dehydration synthesis. So remember, we talked about this last week. Dehydration synthesis is when you lose a water molecule while joining two monomers together. So we will be talking about dehydration synthesis again today, and a little bit about, oh, I'm blanking on the other one. Okay, what's the opposite, guys? What's the opposite of dehydration synthesis? Anybody remember? When you break, oh, I remember what it is, but I want to see if you guys remember. Anybody?
Hydrolysis.
Joshua got it. Hydrolysis, that's right. Hydrolysis is the opposite, where you split a water molecule to separate two molecules. Sometimes I get myself confused. Okay, we'll continue on here. I just have to hit the right button. Okay, so one example of a disaccharide is maltose. This you can find in grain. So my pictures are blinking. So maltose is a glucose molecule, two glucose molecules. Sucrose, this is a common table sugar, glucose plus fructose. So that is another good example of a disaccharide. Lactose you find in milk. Glucose and galactose are the two monomers that are joined together by dehydration synthesis to form a disaccharide. Okay, so just as a refresher of dehydration synthesis, we're looking at the formation of a disaccharide, so glucose plus fructose. So if we back up, glucose and fructose forms sucrose. So what we're looking at is a dehydration synthesis that forms sucrose. So here is our, oh, why am I, there it goes. Here is our H2O. You have the OH right here and the extra hydrogen there. You are basically removing those molecules. You lose water, and then this bond here joins with oxygen. Okay, so we'll see on the next slide. There we go. There's our oxygen that binds those two monomers together to form your sucrose molecule. Okay, polysaccharides. Okay, these are polymers, so many monomers joined together. This is going to be 3 or more monomers, because a disaccharide is 2. So when we talk about polymers, it's 3 or more joined together. And they're joined together again by dehydration synthesis, and this is where your dehydration, you're losing water. So some examples, glycogen. You find these in animal cells and our cells, and this is many monomers of glucose joined together. This is an energy storage molecule. So for example, if you didn't have enough blood sugar, or you didn't have enough sugar in your blood or glucose in your blood, your pancreas would release glycogen, which would basically tell, is it glycogen? I'm getting myself, it would release a hormone. Let's not get too tied up on what hormone it is right now. It would release a hormone that would tell glycogen to release those glucose molecules into the bloodstream so that your blood sugar levels stay constant. So it's an energy rich storage molecule. Starch is from plant cells. And this again is where you have many monomers of glucose joined together. So some examples of starchy things like potatoes, jicama. I'm blanking on starchy things right now. I want to think potato chips. Potatoes are a big one. Okay, so this is what glycogen would look like. We have individual glucose molecules all joined together by dehydration synthesis. And note that the actual polymer is very branched, okay? It's not just a long single string, it's branched and branched and branched. So that is what glycogen would look like in the cell. Okay, now if we do the opposite and we're breaking down sugars, this is where we're using hydrolysis. So remember, hydro means water. Lysis refers to burst or break or split. So this is where we are basically splitting water, and we use that to split apart or break a chemical bond between two molecules, okay? So here we have sucrose, which is it glucose and fructose together, okay? Where we are using water through hydrolysis, we split and we break that bond. And the components of water right here are used to separate those molecules. So now you have two separate molecules. You have glucose and fructose. And the water has been split and is no longer available. Okay. So here's just another illustration of the same chemical equation, but it's all on one page. We have our sucrose, and through hydrolysis, we use water, we split water and break that bond, okay? And now you end up with two separate monomers again. So essentially, which is really cool, we have dehydration synthesis and hydrolysis, which are opposites of each other. You can go back and forth between breaking bonds and building bonds using these two processes. So it's really important that you remember the difference between dehydration synthesis and hydrolysis, because I will definitely ask you questions about that, okay? All right, moving on to proteins. These are the most abundant organic molecules. So you are made of proteins. Your muscles are protein, you have protein fibers, collagen in your bone, in your skin. You have protein everywhere, okay? So protein is a really important molecule, plays a lot of important roles in the human body. So it does have basic elements or components of protein. You will find carbon, hydrogen, oxygen. Anybody want to guess what the fourth one is? Because we talked about the four major molecules that we find in the human body. Okay. Oh, I just missed the message. Nitrogen. Let's see, everybody got it? Yes. Thank you, Taylor, and thank you, Kelsey. You were right. Nitrogen. So these four components are essential to find in protein. Okay, whoops. How do I get this going again? There we go. There are 20 amino acids that combine to form proteins, or we would also call them polypeptides. So that term poly comes up a lot. These monomers bind together, and the bond that holds these proteins together are called peptide bonds. The only time you see a peptide bond is with a protein. So don't get confused if I ask you questions about different types of bonds. Protein bonds peptide bonds are only when we are talking about proteins. Okay, so looking at the structure of individual amino acids first, each amino acid is made up of a carbon atom in the center, hydrogen atom. So you can see in our picture here, we have our carbon. Here's our hydrogen. Then we have an amino group. And so when we have that term amine or amino, that refers to the fact that there is going to be a nitrogen there, okay? So here is our amino group. And then lastly, we have our carboxyl group. And it's kind of fun. I like how it's written out, COOH or COO, one way to remember it. So here is our carboxyl group. So you have these main components for each amino acid. You always will have a central carbon atom. You will always have an amino group. You will always have a hydrogen atom. You will always have a carboxyl group. Lastly, you have a variable side chain, or the R group. And this is what makes amino acids different from each other, is this side chain varies. Okay, so let's talk about protein synthesis, linking amino acids together. And I was going to ask you what type of bond, but it says it right down here. When you join two amino acids together, they are held together by a peptide bond. So linking them together, we use dehydration synthesis again. So we are losing a water molecule, out goes the water. And that joins those two amino acids together at that, between the amino group of one and the carboxyl group of another. Oh, there, said it right there. So the amino group of one amino acid and the carboxyl group of the other join together to form a peptide bond. So we call peptides, this is when you have two peptides is when you have multiple peptides joined together. A dipeptide, it refers to the fact that you only have two amino acids joined together, okay? That would be a dipeptide. What would we call a peptide that is 3 or more amino acids joined together? I'm gonna wait to see if you guys respond. Remember the kitty I talked about? Nobody? Okay, we would call it a polypeptide, okay? Poly meaning many, peptides, many amino acids joined together would be a polypeptide. So 3 or more would be a polypeptide. There we go. These would be tripeptides or even larger. So here we go, showing you kind of the conversion between dehydration synthesis and hydrolysis. They are reversible reactions. Also, it's important to remember. So here we go. We can see at the top, we are starting with dehydration synthesis. We want to join these two amino acids together, the glycine and the alanine, sorry. So carboxyl group on one side here, and the amino group on the other side. Join together, we lose water. There's our water over here. It's been lost. And you join together, and the carbon from the carboxyl group bonds with the nitrogen of the amino group of the other amino acid. And this bond is a peptide bond. Okay. I think we already looked at the protein shape video. I kind of didn't like it. I'm going to see if I can find us some better videos moving forward. There are a lot of great videos online. Oh, speaking of videos, this lecture is being recorded. Once it's converted, I will upload it to YouTube, get the link, and post the link on Canvas. You can find it under the module section, and I will have a whole separate module just for Zoom videos that will be dated for the day that we had the Zoom video, and I'll try to put the topic that we covered as well. So you'll be able to go back and watch the lecture again for studying purposes, which I would recommend. Okay, Protein shape, primary structure. This is where you just have those amino acids all kind of lined up in a chain as a polypeptide. I often refer to them as like pearls on a string. Each little, for example, each little pearl is a peptide, kind of like this. That would be a polypeptide. So each little pearl is an amino acid. Okay, that would just be a simple structure of a polypeptide, the primary structure. The secondary structure, this is where you have hydrogen bonds and you are forming spirals like this, or those pleated sheets that we talked about before. So kind of draw it out if I can. I know it looks funny, but kind of like those fans you made when you were little, you fold them together so you can fan yourself. That would be a pleated sheet. These are two-dimensional and they're held together by hydrogen bonds. So in peptides or polypeptides, you have the peptide bond holding the amino acid together, and you also have hydrogen bonds holding the polypeptide into a specific shape. Hydrogen bonding is really an important bond. It has a lot of important functions. Okay, tertiary structure. This is where you have it folding into a three-dimensional shape, and it coils, it folds, and it's held together at specific points by hydrogen bonds again. It's really important to know and remember that the shape of a protein determines its function. So the three-dimensional shape is incredibly important. If the shape is wrong, the protein will not function. It will be non-functional, and you can have, that could be a disease in the human body. So it's really important that that shape is just perfect. And we'll talk more about why that is in a bit when we talk about enzymes. Okay, quaternary structure is when you have two or more tertiary structures joined together and they interact. And so you have a large macromolecule that is formed. So a great example of quaternary structure of a protein that has this is hemoglobin, which is made up of 4 individual subunits. Okay, so here we go. Primary structure. See how they draw it in circles, kind of like pearls on a string. Easy way to remember it is just single strand. No branching, no coiling, no sheets, no pleated sheets, just a single strand. Okay, here we have a picture of what the secondary structure would look like. You have what the helix, the coils, we call it the alpha helix. And then the beta, that's what the B, this is B means beta. Okay, beta pleated sheets. And so this A, that's A, so alpha. So this is our secondary structure. And remember, they're held together by hydrogen bonds. I'm sorry, my handwriting is terrible today. It's kind of hard writing upright. Okay, then we have our tertiary structure, where we have those pleated sheets and those coils kind of all folding in together and held again by hydrogen bonds. Okay, so now you have a three-dimensional shape. I'm sorry, I got an itch. Three-dimensional shape held together by hydrogen bonds, which some proteins can be done at this point. Others are going to join together to make a macromolecule with a tertiary structure. So again, this shape that you see is very important and very specific for its function. And then here we have an example of quaternary structure. This is a hemoglobin molecule, and each individual quaternary level has an iron molecule or a heme group, which we talked about briefly before, binds to oxygen, and this is attached to your red blood cell. So this is how oxygen travels, or is attached to your red blood cell and travels throughout the bloodstream to the tissues that need the oxygen. Okay, here we have all of our structures together in one place. This is showing the molecular version of primary structure. Each carbon represents an amino group, okay? Then we have, this is our alpha coil, held again by hydrogen bonds. Tertiary structure, so again, we're using hemoglobin as an example. Tertiary structure, you have a three-dimensional shape. And then you have quaternary structure where you have multiple tertiary level structures joined together to form a very specific three-dimensional shape. That shape is incredibly important with its function. And I know I've said it like 3 times already, but it's because it is that important that you remember. Okay, enzymes. Enzymes are proteins, and they have specific shapes. Their shape determines their function. If their shape is off just a little bit, they will not be able to work. We talked before about how enzymes help lower activation energy of a chemical reaction or a chemical process. So if an enzyme doesn't have the right shape, or it's just misshapen just a little bit because maybe you had a mutation and one amino acid is wrong, that could change the entire three-dimensional shape. That enzyme will not work, and you cannot do that chemical reaction that you want to do in your body. Okay, so reactants, which are the substances that react with an enzyme, we call those substrates. We can call them substrates or reactants, okay? So just looking very briefly here, we're not going in depth right now with enzymes, but looking briefly, notice how this enzyme here, catalase, has a three-dimensional shape, and how the reactant also has a specific three-dimensional shape. If those two molecules fit together, kind of like puzzle pieces where it has to be the perfect fit, if they fit, like shown in this picture here, that allows that reaction to occur. And in this example, it breaks apart this H2O2 molecule and then releases them. The neat thing about enzymes is that they are reusable. You can reuse them over and over and over again. As long as there is a substrate available and the enzyme is available, the reaction is going to occur. Your body has the ability to regulate how many enzymes are available at any time, which controls the rate of chemical reactions, which again, that will come up more later as we continue on. Okay, cofactors and coenzymes. We're not going to go too in depth with this at the moment. We're just going to talk about what they are right now. Cofactors play an important role in the enzyme being able to do its function. So how the enzyme is able to work. Some examples of cofactors: calcium ion, magnesium ion, copper ion, and zinc ion. These are all cofactors. These are important ions that your body needs in order to function. So I've already talked about how important calcium is, for example, in the bone, keeping your bone strength up. We talked about the parathyroid hormone. When we were talking about homeostasis, calcium was an important factor in that equation. But you also have other important elements as shown here. Vitamins and minerals also act as cofactors. So making sure you get those trace minerals in your diet, making sure you're getting enough vitamins, either supplementing with vitamins or getting them through your diet, are really important in how your body is able to function. Okay, coenzymes. These are molecules that pick up and carry hydrogen molecules. They play important roles in certain chemical reactions. Specifically, we're going to spend time talking about coenzymes a lot when we talk about metabolism. So that's just something you have to look forward to. So over here, This is showing you cellular oxidation, which is part of metabolism. But all of these, so examples would be NAD or FAD. When they pick up a hydrogen molecule, it becomes NADH or FADH2. Okay, so they're hydrogen molecule acceptors. They carry them. We'll talk about why that is important later. You don't need to worry about that now. When we talk about metabolism, you will need to know all of the details. Yay, something to look forward to. Okay, lipids. Lipids are pretty much hydrophobic molecules such as fats, oils, and waxes. These are going to be, so remember what hydrophobic means, they repel water. So they are insoluble with water, they cannot dissolve into water. So remember, if you pour oil into water, you're going to get two layers, one layer of water and a layer of oil on top, because they don't mix. They cannot dissolve into each other. Oil repels water. So some examples of lipids. Glyceride is a fat that you find in animal bodies. Phospholipids, we've briefly talked about the importance of phospholipids, and we're going to talk more about them. Glycolipids, I can't say this one. Eicosanoids, so forgive me if I was wrong. Eicosanoids, there we go, eicosanoids. And steroids, these are all lipids. So first, let's look at a glyceride molecule. A glyceride molecule is made up of two major components, a glycerol molecule and a fatty acid. When you join those together, you form a glyceride. A very good example of a glyceride is a triglyceride. I'm sure you've heard about those before. We talk about them sometimes when we talk about your diet and how much you should have in your diet, triglycerides. A triglyceride, tri meaning three, means you have three glyceride or glycerol, sorry, three glycerol molecules. So here is our glycerol molecule. There's three of them, and they join together with a fatty acid. So here's our fatty acid. They join together via dehydration synthesis, and you end up with three fatty acids attached to three glycerol molecules, making a triglyceride, plus the 3H2O that was removed in the formation of dehydration synthesis. Okay, sorry, I can't see my title. Give me one sec. Glycerides. So still talking about glycerides. So fatty acids attached to a glycerol molecule. Triglycerides, this again is a glycerol molecule plus three fatty acids. That means tri means three. There are three important functions that you need to know about triglycerides. First, it is an energy source. Sorry, I'm going to take a sip of my coffee. It is still morning. Okay. Second, insulation. So we talked about how fat has important functions. Triglyceride is a fat, and one of the functions is insulation, insulating organs, insulating tissues, very important role. So there you need a certain amount of fat to be healthy. And then protection. So you do have organs that are surrounded by fat, and a certain amount of that is necessary. It performs protection, cushioning. So if you are hit in the stomach, you have a sheet of fat that covers your intestines called, oh my goodness, greater omentum. And that helps to protect your organs. You also have fat that encases each kidney, which acts as protection as well. So just examples. You even have fat surrounding the heart. Now, how much fat can be a problem? Again, there's a certain amount that's healthy, And then if you have too much is when your body goes out of homeostasis. Okay, so looking again at this example, dehydration synthesis, forming a triglyceride from 3 glycerol molecules and 3, oh my gosh, just 3 fatty acids. Okay, so again, this reaction can be reversible. And when you reverse it and you break down a triglyceride back to its smaller subunits, you are doing it through hydrolysis. Okay, so looking at fats. Fatty acids can be saturated with hydrogen. And what this means is that if we look down here at this picture, see how each carbon, there's single bonds, and each carbon is either bound to another carbon or a hydrogen molecule. So each carbon is saturated with hydrogen. Every single available spot is taken by hydrogen. That actually plays a role in how the fatty acid behaves. So because it is saturated with hydrogen, there are no double bonds, which is really important to remember. Saturated fats have no double bonds. And what that does is that these molecules tend to be flat and they stack on top of each other and they condense really well. And so what happens if we look over here, coconut oil and palm oil, for example, are saturated fats. At room temperature, they are solids. That is because they are saturated and because of the flat shape of the molecules stacking on top of each other, okay? No double bonds. Unsaturated, on the other hand, has 1 or more double bonds in that fatty acid tail. This means it is not saturated with hydrogen. You have less than the total amount of hydrogen that you could have. So examples, mono unsaturated. Mono refers to 1. So this is where you have 1 double bond in the fatty acid. And every time you get a double bond, that chain bends. So you don't have a flat shape anymore. And the more double bonds you have, the more bent that molecule is. And so they're not going to be able to stack on top of each other nice and neat like saturated do. They're bent, they're jumbled, and that gives them the property of at a room temperature being a liquid. Because you have those double bonds, they cannot stack on top of each other the way it would be if it were saturated with hydrogen. Polyunsaturated is when you have, so poly means many, 2 or more double bonds. So if you look at the health contents on a, let's say you have a drink, let's say you have a Starbucks double shot, which just so you know, I'm going to talk a lot about coffee this semester. I did used to work at Starbucks, so it's kind of ingrained into me. So if you're looking at the contents on the back, it will tell you saturated fat, unsaturated fat. That is what it means. So when you talk about saturated and unsaturated fats, you now know what it means when we talk about the structure, how the molecule behaves. And typically, let me ask you, which one is a healthier type of fat? A saturated fat or an unsaturated fat? I'd like you to respond in the chat, please. Okay, Jimmy, you're right. Unsaturated is healthier for you. Can you think of any other examples of saturated fats? Typically, most often, it's going to be from animal products. Okay, there are a few products from plants like we see here, the coconut and palm oil, but typically saturated fats are coming from animals. Yes, Ava, butter is a great example of a saturated fat. Down here we have a picture of the unsaturated fats. Can you guys think of Science kind of touts a specific type of unsaturated fat as being really healthy for you. Can you think of any examples other than what you see in the picture that is an unsaturated fat that's considered to be healthy? It's used a lot in cooking. Yes, Ava's right again. Olive oil was what I was thinking about. Olive oil is one of those really healthy, unsaturated fats. Another great one is avocado oil. Also an unsaturated fat. It's a liquid at room temperature. And also, just so you know, avocado oil has a high burning point, so it's great for cooking as well. Just a little FYI. Okay, we will move on. Maybe. There we go. So here we're looking at a simple fatty acid, a difference between unsaturated and saturated. And so this would be monounsaturated because there's just one double bond between those carbon molecules. Down here is saturated, fully saturated with hydrogen atoms. Okay. Now we're going to move to phospholipids. So phospholipid is made up of three sections, but we usually just break them down into two. So we break it down to the head of the phospholipid. Here we go, the head, and then the tail. So if we look specifically at one phosphate or phospholipid, it is made up of a phosphate group linked to two diglycerides. Okay, so there's our fatty acid chains right here. There's two of them. And we have a glycerol backbone, because remember, fatty acids attached to glyceride. And then we have our phosphate group, okay? And this molecule is a polar molecule. Do you remember what polar means? Polar means you have a charge difference across a molecule. So it's not completely neutral, for example. So what happens is this phosphate group actually gives it a negative charge. And so the head of the molecule is attracted to water because water is also polar. But we have this fatty acid tail, which is a lipid, which repels water, doesn't want water around it. So the way you find phospholipids in your body is as a two layers. We call it a phospholipid bilayer. Okay, that is our official term for what we see right here, phospholipid bilayer. So extracellular versus intracellular, we're just looking at a little section of a plasma membrane. So phospholipids combine to form two layers which make up the plasma membrane of a cell. So those phosphate heads face outwards towards the water-soluble extracellular fluid, and then those heads face inwards towards that water-soluble intracellular fluid. So you end up with this sandwich of heads, and then these fatty acid middle of the sandwich, let's say your peanut butter and jelly, and then another layer of heads. So this helps with the formation of cell membranes. It is a really important function. We'll talk more about cell membranes as we continue, but knowing what a phospholipid is, how it behaves in water, for example, and how it repels water with the fatty acid tails, that's why they join together to form this sandwich-like structure. Okay, glycolipids. A glycolipid is a glycerol molecule, two fatty acids, and one carbohydrate. So glycolipids have a carbohydrate attached to them. So let's see, here is a glycolipid. You're going to find them embedded in the plasma membrane of a cell. Okay, so you'll have these phospholipid, here you can see your plasma membrane. The blue circles are the phosphate heads. Then you can see the little squiggly tails. So if the tail is squiggly or bent, what does that mean? Would we consider it saturated or unsaturated? Let's see. Unsaturated, that's right, Jimmy. When it's bent, that means it's not laying flat on top of each other, and that's really important in how the cell membrane functions. If they were saturated, they would be smashed together so tight, there wouldn't be any fluidity to the plasma membrane. And it is fluid. It moves, it's flexible. So you don't want them to be smashed tightly together. So you want those bends in it, okay? Sorry, I know I got off topic. But in the plasma membrane, you have glycolipids that are embedded in there. Those carbohydrates that are attached extend out to the outside of the cell and play important roles in how the cell recognition, basically cell recognition is the big role of that. Okay, so you find these in the plasma membrane. You also find other molecules embedded in the plasma membrane, like cholesterol. I know I'm jumping ahead about cholesterol, but cholesterol also plays a role in the fluidity of that plasma membrane. Okay. What are we looking at here? Okay, we're still doing lipids. Okay. Oh, the word I can't pronounce. Eicosanoids. These are made from fatty acids called arachidonic acids. I'm going to be honest, we're not going to talk a lot about eicosanoids, but it is an important fact for you to know. An eicosanoid, an example of it, I need to work on this pronunciation, the eicosanoid, and a good example of an eicosanoid is prostaglandins. So I take it back. You do need to know what prostaglandins are, okay? It is an important local hormone. It's made of short chains of fatty acids, and prostaglandins do play an important role. They stimulate nerve endings, and they give you that sensation of pain. So if you're hurting, that's one of the reasons that you continue to feel pain, is that these prostaglandins are stimulating those nerve endings and triggering that signal to your brain, which makes you feel the pain. So darn those prostaglandins, right? Another important role is they can help trigger labor contractions. As the cervix ages and starts to stretch, when that fetus is getting to be full term, it starts to release prostaglandins, which can trigger contractions. Interesting fact, you can also find prostaglandins in semen. So doctors will tell pregnant women who want to go into labor, want to get done with being pregnant, to go home and have intercourse with their partner to help try to trigger those contractions with the presence of semen. So kind of a little factoid. We'll talk about that again when we talk about the reproductive system, which is at the end of the semester, but wanted to add that as to one of the functions, or where you find that where you find that in the body. Okay. Steroids. Steroids are made up of 4 rings of carbon. They are hydrophobic, so they're lipids, they're hydrophobic, and they have different functional groups which determine their function. So 3 6-carbon rings, 1 5-carbon ring, joined together to make this 4-ringed carbon structure. Then there are side chains, just like when we talked about amino acids that has that that side functional group, steroids also have a side functional group that attaches to the ring that determines its function. So some examples include cholesterol. Okay, and I already mentioned this, but cholesterol is an important component of the plasma membrane, of the cellular membranes, helps to lend to the fluidity of that plasma membrane. Okay, another example of a steroid would be sex hormones like estrogen and testosterone. Estrogen and testosterone, just so you know, can be found in both males and females, just the level of it varies. Obviously, more estrogen in females and more testosterone in males. Steroid hormones like calcitriol, which is vitamin D. And vitamin D, we will talk about a lot when we talk about calcium regulation, but it does play a really important role in your intestines' ability to absorb calcium from your food into the bloodstream. If you don't get enough vitamin D, you cannot absorb the calcium N, and that affects your blood calcium levels, which can in turn affect how much calcium is in your bone, because your bone is your calcium storage area. If you don't have enough calcium in your blood, it's going to take it out of your bone. So if you are chronically low on vitamin D, that's going to affect your bone density and strength. So get your sunshine or take your vitamin D and your calcium just to be safe. Okay, looking at these examples, cholesterol, here's that different functional group. With estrogen, that functional group is just a hydroxide. And with testosterone, two hydroxides. With testosterone, the only difference between testosterone and estrogen is estrogen has a hydroxide group here, and testosterone has an oxygen. So they are very, 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 very similar. And they play almost similar roles in the female body versus the male body, which we'll talk about when we go into the reproductive system and when we do endocrine system. Okay, looking a little bit more at cholesterol. Remember, all of these molecules we've talked about, the steroid hormones, calcitriol, and cholesterol, all are these four-ringed carbon structures. But you find, for example, you find cholesterol in the plasma membrane, helps with that fluidity, and it is an important role. Okay, let's see. We're going to stop for today. I know it's a little early, but I want us to have a little bit of time to talk about nucleic acids next class period. But before I leave, I want to open it up for questions. Are there any questions you have? Just unmute yourself and pound away, or you can send a message so I can help you out. Let's see if I can make this bigger. There we go, bigger. Any questions about what we talked about today or with the course? Anything about that? We're feeling good? Okay, if you're not comfortable asking right now, that's totally fine. You can text me or send me an email and I will get back to you as soon as I can. Just remember, you have those resources available to you all the time. And if you need me quick, text me. Okay, so question. So unsaturated is the bent bending fatty acid. Yes, that is when you have one or more double bonds. That double bond causes the bend. Okay, that gives that physical shape, which lends it to that more fluid structure. Good question, Drew. Any other clarification points? Okay, I will let you guys go for now. Give me 1 second. I'm going to grab our schedule And we'll look at that for 1 sec. Oh. Okay, so looking at our schedule, let's see. So we are actually ahead, a little bit ahead. So we already did our chemistry. We're almost done with macromolecules. So we're gonna spend some extra time with macromolecules on Thursday, try to get some videos that are helpful to kind of visualize some of these things. So we're on track, we're doing good. Make sure that you're working on your workbook homework. So that is on Top Hat. So you wanna make sure that you get that done. Those due dates are on Top Hat. So it's really important that you check that regularly. Don't wait till the last minute, please. Try to work on them as we do it in class, work on them or work ahead so that you're prepared for the class period, which is actually a really good idea. In our schedule, it says there's an organic chemistry worksheet. We don't have one, so you can disregard that, okay? So I'm gonna cross it out on my list. So disregard the organic chemistry worksheet. If I do get the worksheet from another professor, I will assign it, but I will let you know. But for now, do not worry about that worksheet. You have your workbook on Top Hat that gives you this work and gives you the homework and helps you review, which I think is going to be very beneficial. So use that as a tool for your learning. And just as a reminder, for Canvas, that's where you find your quizzes, your homework, and your course content like PowerPoints and your Zoom lecture links, and your grades. Grades will be updated this weekend with your new homework and your quiz scores and all of that. So expect by Sunday, I know it's a little late, but if I can do it sooner, I'll send an update to you via email. But expect that your grades will be updated with your workbook grades and all of that into Canvas, because I have to transfer everything from Top Hat to Canvas manually, which really stinks. So that's going to take me a little bit of work, but I will get that done this week for you. Oh, question. Where do you find Top Hat? Okay, not sure how to do it. I got the code, but don't know how to use it. Okay, so let me see if I can go to our syllabus. Let me share my screen. I need to share my screen first, sorry. Internet. Okay, I need to move this. Give me a sec. This thing is in my way. Okay. Oh, I'm looking at the wrong thing. Okay, no, I was looking at the right thing, sorry. Okay, so if you go to Physiology, to your course on Canvas, go to Modules, click on that, and then go to Syllabus. Click on the syllabus. That will load in a moment here. And then you just scroll down right there where it says required study materials. It gives you the course URL. So you just use that link. Go to Top Hat. This is the join code for our course. Now, if you've already tried to join and you cannot join, email me because I may have to individually invite you, okay? If you have the code from the bookstore and you still haven't used that, that code is your password that you use when you are logging in. Let's see, we have somebody responding. Okay, what are we supposed to do with mastering? Nothing. You do nothing with mastering biology. That shouldn't, is it even available for you to see? Let me see. Oh, I have so many things on my screen right now. Okay, there's nothing that you do with mastering biology. That is for when we are not in a COVID crisis. So we are using Top Hat for all of our course homework. Nothing with mastering biology, okay? Are there any other questions? Drew says no. I like your cat, Drew. What's your cat's name?
It's Nellie, but it's my roommate's cat.
That's okay. It's your cat by proxy. Nellie.
Pretty much.
Yeah.
She loves my dogs, so she always hangs out with me.
That's awesome. You will probably hear my dog at some point. I have a long-haired Chihuahua who likes to bark when the mailman comes, so he's a character.
Yeah.
Pets are
I had to make sure I was muted.
I can't be muted, so you're going to hear it at some point. Just telling you now. It's going to happen. Okay. Any more questions before we go for today? Okay, I'm going to stop that share. Okay, it looks like we're good. It's going to take some time for me to convert this video to something that I can upload onto YouTube. So look for the video to be available tonight for studying, okay? All right, I hope you all have a fantastic day. Wish me luck with that COVID test because it's not going to be fun. And I will talk to you on Thursday. And if you have questions in the meantime, you know how to reach me. All right, bye.
Are we doing Zoom on Thursday?
Yes, I will send a Zoom link.
Okay.
Yeah, very good question.
Okay.
We will be doing Zoom. All right, have a good day, everyone.